This video provides an introduction to the topic of cellular communication or cell signaling, and it will address the following questions. Why is it important for cells to send and receive signals to and from the outside world? What is the nature of the signal? What are the main types of receptors cells use to detect chemical signals? What intracellular signaling pathways do these receptors activate? What are second messenger molecules and what is their role in intracellular signaling pathways? What nuclear and cytoplasmic cellular responses can be elicited? How do cells turn off the signal? And why is it important that they do so? So why is it important for cells to send and receive signals with the world outside the cell? If we think about this in the context of multicellular organisms like animals and plants, it's apparent that the organism as a whole can only function due to the integrated activities of the various specialized tissues it's made up of. Well, how do those activities get integrated? Via communication between the hundreds of different and specialized cell types that make up the organism. In an animal like us, cellular signaling is used for control of various functions by the nervous system, to communicate information about nutrient status, to stimulate growth and development, and so on. Virtually every aspect of our anatomy and regulation of our physiology depends on cellular signaling. But all organisms need to be able to detect and respond to important stimuli in the environment, and the types of stimuli that can be used to initiate a cellular response are incredibly diverse. They include light, which initiates signaling in cells in our retina, which then gets communicated to the central nervous system and perceived as vision. Plants also detect and respond to light-dark cycles and regulate their growth patterns accordingly. And there are lots of other examples of how light can be used as a signal. Cells can also be sensitive to mechanical stimuli. This is the way that hearing works. There are specific cells in the inner ear that are stimulated in response to sound waves, which are essentially pressure waves. It's also how you detect tactile stimulus or touch, via the stimulation of specific touch-sensitive neurons in your skin. Likewise for detecting changes in temperature via temperature-sensitive neurons in the skin. Changes in voltage potential due to the movement of charged ions can also be a signal that certain cells are responsive to. In fact, this is an important signaling mechanism in the nervous system. The most prevalent stimulus that all cells respond to in one form or another is chemical signals. Listed here you see some examples of the diversity of these types of signals, which includes nonpolar gases, lipids like steroid hormones or hormones derived from membrane lipids like arachidonic acid, which is important for pain and inflammation signaling, nucleotides like cyclic AMP, and very, very commonly, signaling proteins such as insulin. The responses that these cellular stimuli can elicit in cells are diverse and highly specific for whatever signal and cell we're looking at. Often, the signal causes a change in gene expression, which leads to the repression or activation of production of new proteins. This is a relatively slow response to see implemented, occurring on the order of minutes. Often, a cellular stimulus leads to changes in the regulation of enzymes already present in the cell, either activating or inhibiting their catalysis. And this is a very fast, virtually instantaneous response that almost immediately changes the biochemistry happening in the responding cell. Sometimes the stimulus leads to the fusion of vesicles already present in the cell with the plasma membrane, which either releases the contents of the vesicle to the outside world or makes the vesicle membrane, along with all of its embedded proteins, now a part of the plasma membrane, which changes the composition of the plasma membrane and therefore alters the cell's interaction with the outside world. Sometimes the signal leads to a reorganization of the cellular cytoskeleton and growth or motility of the cell, or a change in the shape of the cell, as we see with muscle cell contraction. Finally, the most extreme cellular response that might be stimulated is apoptosis, or programmed cell death, in which a cell initiates a signaling cascade that ends up fragmenting and destroying the cell. This is an important control mechanism in multicellular organisms to remove unneeded or sick cells. So how do these diverse signals influence a cell's behavior in all these interesting and varied ways? Via interactions with cellular receptors, which in turn interact with and stimulate various intracellular molecules, mostly proteins, to cause a specific cellular response. In this video, you'll be introduced to some of the most common classes of cellular receptors. In the next video, we'll look at some specific examples of interesting and biologically relevant signaling pathways that use these important receptor classes. Most signaling receptors are embedded in the plasma membrane, which makes sense since they are sensing stimuli present in the outside world or responding to chemical signals, which are primarily polar molecules that can't pass through the cell membrane, but rather are stopped by the barrier function of the plasma membrane. An exception is lipophilic molecules like the steroid hormones, which are nonpolar and small enough to slip right through the phospholipid bilayer, and as a consequence, their receptors are actually located within the cytoplasm of the cell. Steroid hormones like estrogen, testosterone, glucocorticosterol, and so on all interact with cytoplasmic receptors present within their specific target cells. Steroid hormones nearly always act to alter gene expression in their target cells, and the receptor is actually a transcription factor, that is, a protein that interacts with DNA to activate or repress the expression of specific genes in the genome. Often, in the absence of the steroid signal, these receptor transcription factors are sequestered in the cytoplasm by chaperone proteins that prevent their passage through the nuclear pores. When steroid hormone binds, these chaperones dissociate, and the hormone receptor complex translocates to the nucleus. In some cases, the receptor is actually located within the nucleus, and the steroid hormone meets its receptor there. In any case, once in the nucleus, the hormone receptor complex binds to specific DNA sequences found in target genes and alter the expression of these genes, either up or down regulating them. You can see an illustration of a specific steroid hormone receptor, actually a paired dimer, binding to its recognition sequence in the DNA, sometimes referred to as a hormone response element. If the effect of receptor binding is to activate gene expression, then more mRNA will be copied from the gene. And after export from the nucleus, these mRNAs will be translated into proteins by ribosomes, resulting in an increase in that protein's expression in the cell, and vice versa. Down-regulating an mRNA expression results in less of that protein in the cell. This is a relatively slow response. It takes minutes to transcribe and translate new protein. Other types of cellular receptors trigger signal transduction pathways in the cell that lead to very rapid changes in cellular metabolism and physiology through the regulation of proteins and other molecules already present within the cell. Most cellular receptors are embedded within the plasma membrane and transduce the signal from outside world to inside world by activating or inhibiting membrane-associated or intracellular proteins or other molecules. We'll now look at three specific classes of cell surface receptor that act in this way, beginning with gated ion channels. As the name suggests, these receptors are channels in the membrane that control the passage of specific charged ions. These channels are special in that they change shape to either allow or prevent the passage of ion, depending on whether their signal is present or not. Signaling by these types of receptors relies on previously established concentration gradients that have been created via active transport. For example, sodium and potassium gradients created by the sodium-potassium pump. This type of signaling is analogous to the idea of a locked gate in a fence. If you've got the key to open the gate, you can pass through. If not, you're stuck on the outside. In this case, the key is the signaling molecule or ligand, and what we're looking at is a ligand-gated ion channel, a channel whose gate is opened or closed in response to a chemical signal binding to the extracellular domain of the receptor. Ligand binding induces a shape change in the receptor that opens or closes the gate, represented here as this little flap. But more realistically, the shape change causes the pore the channel makes through the membrane to either open or close, depending on the signal and depending on the receptor, which we can see here in this top-down view of the acetylcholine receptor, which is the receptor that mediates muscle cell contraction in response to nervous system signaling in animals. More on that receptor in the next video. Gated ion channels can be gated by other types of stimuli besides chemical signals, including mechanical stress, temperature, and voltage changes, and we'll see an example of that as well. A second type of membrane-embedded signaling receptor is the G-protein coupled receptor. And again, the name very nicely describes how this receptor works. This is an extremely common receptor class present in all eukaryotes, and it's an important class of targets for pharmaceuticals. More than half of our clinical drugs target G-protein coupled receptors or their signaling pathways. This type of receptor physically interacts with an intracellular protein called a G-protein, hence the name G-protein coupled receptor. This intracellular G-protein is called a G-protein because it binds to and hydrolyzes GTP, guanosine triphosphate. It's often referred to as a heterotrimeric G-protein because it consists of 3 protein subunits, alpha, beta, and gamma. Most commonly, when bound to an intact GTP molecule, the G-protein is activated. Once the GTP is hydrolyzed to GDP and phosphate, the G-protein returns to its inactive state. So G-proteins are signaling proteins that are also enzymes, and importantly, because of this enzymatic activity, they effectively are able to switch themselves off, thus terminating the signal. Once in their activated state, G-proteins activate other proteins, often enzymes, which leads to some specific downstream cellular response depending on the cell type and the downstream pathway that gets triggered. The steps that take place in response to G-protein coupled receptor activation follow this characteristic pattern. First, ligand binds to the G-protein coupled receptor, or GPCR. This causes an inactive G-protein, which was bound to GDP, to release that GDP and bind a fresh new GTP molecule, which results in activation of the G-protein. The now activated G-protein becomes mobile along the membrane and slides away from the receptor, interacting with and activating, or sometimes repressing, the activity of a second enzyme. The target enzyme initiates some specific cellular signal transduction cascade that results in a particular response. The G-protein then hydrolyzes its GTP to GDP and phosphate, thus inactivating itself until the next receptor signal is received. The target enzyme returns to its inactive state without the interaction of the G-protein, and both of these are important ways of terminating the signal. You'll notice in the diagram that the ligand just went away, and you might wonder what happened to it. Cells have several ways to terminate a signal at the level of ligand-receptor interaction, and we'll talk more about these towards the end of the video. The final class of receptor we'll see in this video are the receptor kinases, which, as their name implies, are receptors that also have the enzymatic activity of a kinase. Kinases, as we know, catalyze phosphorylation of target molecules. All organisms have versions of receptor kinases, but one family that has evolved relatively recently and that has really important clinical importance in humans is the receptor tyrosine kinase family. These are so named because the kinase activity phosphorylates specific tyrosine amino acids in target proteins. There are more than 50 of these receptors encoded for in the human genome, and they are often responsible for mediating growth factor signaling pathways that regulate growth, development, and cell division. Given that cancer is characterized by abnormal cell division and loss of normal cell growth controls, you might guess that receptor tyrosine kinases could be involved in the development and progression of cancer, and you would be exactly right. Abnormal activation of these growth factor receptors can result in high levels of cell division and ultimately tumor formation if the cells are not removed by the body, which thankfully most of them are. Receptor tyrosine kinases have a characteristic activation process that is initiated by ligand binding to their extracellular domain. The first thing to know is that these receptors often require a partner to achieve full activation. When ligand binds to both individual kinase receptors, they associate with one another, forming a dimer. Once associated, the kinase activity of the intracellular domain is activated, and the receptors phosphorylate each other's kinase domains on specific tyrosine residues. This is called cross-phosphorylation or autophosphorylation. These phosphorylated tyrosines are then recognized and bound by intracellular signaling proteins, which bind, are phosphorylated and activated, and then transduce the signal to the interior of the cell. Again, the specific signal transduction pathway that is activated in the cell and the response that gets triggered depends on the specific ligand, receptor, and cell type. In the next video, we'll see a specific example of a histidine kinase receptor signaling in bacteria. Now that we have a better sense of the types of receptors used in cell signaling, let's take a closer look at the commonly used intracellular messengers that relay the signal from the receptor and actually cause a specific cellular response to occur. Very commonly, stimulation of a cell surface receptor activates a kinase cascade within the cell, in which a series of protein kinases are activated, ultimately leading to the phosphorylation and activation of a specific protein that directly causes something specific to happen in the cell. For example, in this scenario, activation of a receptor, and this could be a G protein-coupled receptor or a kinase receptor, leads to activation of an initial relay molecule. This could be an enzyme or an allosteric regulator of an enzyme. And this relay activates a specific protein kinase we'll call kinase 1. Once kinase 1 is activated, it phosphorylates and activates kinase 2, which phosphorylates and activates kinase 3, which in this example phosphorylates and activates the final protein in the relay that is actually responsible for triggering the response. Notice that for each kinase that phosphorylated the target, there's also a phosphatase enzyme, indicated by this little guy labeled PP, that dephosphorylates. So activation of this pathway is going to depend on the relative activity of the kinases and phosphatases in the pathway. Now this seems needlessly complicated. If the response is generated by this final protein down here, why not just have the relay protein activate it directly? Or even better, why not have it directly activated by the receptor itself? There are a few reasons this more complicated signal transduction pathway is needed. First, the receptor is membrane-bound, but the final protein might be located somewhere else in the cell. So we have to have a relay system to communicate the signal from the cell surface to the cell interior or to specific internal organelles. Second, recognize that this multi-step pathway allows for amplification of the signal at each step. This kinase 1 isn't going to only phosphorylate one kinase 2 enzyme. There will be dozens or maybe even hundreds of this second kinase that get phosphorylated, and each of those can then phosphorylate dozens or hundreds of kinase 3 enzymes, which can then phosphorylate dozens or hundreds of these final protein effectors in the pathway. This gives a final response that is orders of magnitude stronger or faster than if the pathway were more direct. The other thing to realize is that cells have thousands of cell surface receptors which are receiving numerous signals from the environment, and all of these signals have to be integrated within the cell. So while this pathway may say to the cell, when ligand stimulates this receptor, generate this response. But there may be another, different receptor and signaling pathway that could intersect with this one and affect its implementation. Just to illustrate this point, let's say this pathway is downstream of a growth factor, and the cellular response that would be generated is cell division and growth. But what if this second receptor was responding to other signals, relaying information about nutrient status? More specifically, that the organism is low on nutrients, low on energy reserves, and running out of organic building blocks that are needed for growth. Can we see how it would be an advantage to have these pathways talk to each other so that the nutrient status signaling pathway could potentially block the growth pathway from going forward if conditions aren't favorable? The more steps in the pathway, the more points of intersection there potentially are with other pathways in the cell, and more opportunities for increased complexity of regulation and integration of signaling pathways and their responses. Multi-step kinase phosphatase pathways are very, very commonly used to transduce cellular signals. Equally common, though, is the use of what are called second messengers. These are not enzymes, though some are the products of enzymes. Rather, they are small, diffusible, non-protein molecules or ions that transduce cellular signals by diffusing through the cell and interacting with target proteins to affect their function. We'll look at three commonly used second messengers: cyclic AMP, IP3 and DAG, and calcium ion. Cyclic AMP is a nucleotide-derived second messenger produced by the action of the enzyme adenylyl cyclase, which converts ATP to cyclic AMP by cleaving off the two terminal phosphate groups and cyclizing the remaining phosphate to the ribose sugar of the nucleotide. Adenylyl cyclase can be activated by a number of different receptors in the cell. Cyclic AMP, once produced, allosterically activates or inhibits specific enzymes, which leads to specific cellular responses. We'll see an example in epinephrine signaling in the next video. Like we saw with the kinase signaling cascade, there's an enzyme that deactivates cyclic AMP called phosphodiesterase, which cleaves the bond between the phosphate group and the 3' carbon of the ribose sugar, yielding plain old AMP, which doesn't act as a second messenger. So if adenylyl cyclase functions as the on switch for this second messenger signaling, phosphodiesterase functions as the off switch, terminating the signal. IP3, which stands for inositol trisphosphate, and DAG, which stands for diacylglycerol, are second messengers produced by the action of the enzyme phospholipase C, or PLC for short. PLC is commonly activated downstream of G-protein coupled receptor signaling pathways, and when it's activated, it cleaves a membrane-embedded molecule called PIP2, for phosphatidylinositol bisphosphate, which is shown here on the left, into DAG, which remains embedded in the membrane, and inositol trisphosphate, which is polar and can diffuse away from the membrane into the aqueous cytoplasm. Both DAG and IP3 can be used as second messengers in specific signaling pathways. Like cyclic AMP, they interact with and activate cellular proteins to stimulate specific cellular responses. We'll see an example of IP3 signal transduction in response to the hormone glucagon in the next video. Termination of IP3 signaling can occur a couple of ways. IP3 can be hyperphosphorylated in the cell to form IP4 or even IP5, after which it is degraded. Or it can be dephosphorylated and recycled to regenerate more membrane-embedded PIP2. Another commonly used second messenger in signaling pathways is calcium ion. We've talked before about how cells can maintain sodium and potassium gradients across their membranes using active transport mediated by the sodium-potassium pump. Well, they do this with calcium as well. Calcium concentrations within the cell cytoplasm are maintained at relatively low levels compared to the extracellular space, the mitochondrial matrix, and the endoplasmic reticulum through the activity of calcium pumps. These pumps carry out primary active transport of calcium from the cytoplasm into these compartments and out of the cell. Because a gradient is established, when a signal is initiated that leads to stimulated calcium release from these compartments, calcium can be used as a second messenger to interact with and affect the function of other cellular proteins and enzymes. We'll see an example of this in the next video as well, when we look at acetylcholine signaling controlling muscle contraction, as well as downstream of glucagon signaling. Because the pumps are always working to reestablish cytoplasmic calcium concentration, that acts as a mechanism for terminating the signal as concentrations are returned to normal. It's important to note that a cell's physiology at any given moment is determined by the sum of cell signaling responses and integration of multiple pathways that are occurring at that moment, as illustrated here. where signals A, B, and C could lead to cell survival. Addition of signals D and E on top of those might trigger growth and cell division. On the other hand, signals F and G in conjunction with A, B, and C might trigger differentiation instead, or specialization of function for this cell. And loss of survival signals A, B, and C might lead to programmed cell death. Researchers have done a really good job of digging down into the molecular mechanisms involved with individual signaling pathways and individual cellular responses. But we've only recently started developing tools, both experimental and computational, to try to tease out all the complicated interactions that are occurring at the same time across the entire cell. This is one important topic in a larger field of study called systems biology, which integrates biology, computer science, mathematical modeling, engineering, chemistry, and physics to try to understand biological function at a holistic level. So obviously, initiating the signal at the receptor is important. Transducing the signal to achieve the needed response is critical. But equally important for the cell is the ability to terminate the signal so that it can continue to respond appropriately to its dynamic and always changing environment and needs. As we've talked about these signaling pathways, we've already discussed some molecular mechanisms for terminating the signal transduction pathway. As we've seen, G proteins have intrinsic enzymatic activity that hydrolyzes GTP to GDP, which inactivates the G protein. For every kinase, there's a corresponding phosphatase that reverses its phosphorylation. Cyclic AMP is cleaved and deactivated by phosphodiesterase. IP3 can be degraded or recycled and reused in the cell. And finally, restoration of cellular ion concentrations by active transport pumps terminates signaling that relies on flux of ions like sodium, potassium, and calcium. What we haven't talked about yet are the mechanisms for terminating the signal at the level of the receptor. Obviously, if the signal is no longer being received, concentrations will fall and eventually will be below a threshold level required to stimulate signal transduction. Since as concentration decreases, receptors are more likely to not be bound by ligand and therefore more likely to be in their inactive state. But cells also have mechanisms to actively terminate the signal by getting rid of the ligand or by removing the entire receptor-ligand complex from the cell surface through endocytosis. There are a couple of ways to get rid of the ligand. First, if we're talking about a scenario involving local signaling, where one cell is releasing a signal to stimulate a nearby cell, the releasing cell might simply take back the signal. This is what we often see with neuronal signaling in the nervous system, which is illustrated here. What this diagram shows is two cells, a neuron here at the top, and then a target cell here at the bottom, which could be another neuron, a muscle cell, or a gland. Neurons stimulate their target cells by releasing stored ligands, referred to as neurotransmitters, through exocytosis. These neurotransmitters then bind to the receptors on the target cell to initiate the response. But the releasing cell often has transport proteins specific for the neurotransmitter, which will then actively transport it back into the cell for repackaging and reuse. Alternatively, what sometimes happens is that the releasing cell or the target cell secretes an enzyme that degrades the ligand here in the extracellular space. In either case, the concentration of ligand is decreased, effectively terminating the signal. The other way that cells can terminate signaling at the level of the receptor is to remove the receptor-ligand complex from the cell surface so that it no longer can activate membrane-associated molecules required for signaling. This is referred to as receptor-mediated endocytosis, and it relies on the existence of intracellular molecules that can associate with collections of activated receptors and cause the membrane to deform, eventually forming a vesicle. The vesicle is then internalized within the cell, fuses with other vesicles to form an endosome, which then acidifies, causing the ligand to dissociate from the receptor. The endosome then sorts the receptors and ligands to separate vesicles. Receptors are recycled to the plasma membrane, and ligand is sent to the lysosome for degradation. One class of these proteins that cause the membrane to invaginate and form vesicles are the plathrins, which have this three-armed structure called a triskelion. In the presence of appropriate adapter molecules, these can self-associate with one another, and when they do, they eventually form these sphere-shaped structures shown here. Now remember that this is happening at the inner surface of the plasma membrane, so what we're looking at in this electron micrograph are clathrin-coated vesicles on the inside cytoplasmic face of the plasma membrane. Once the sphere is fully formed and the vesicle pinches off from the membrane, other proteins stimulate the disassembly of the clathrin coat, and vesicle fusion, endosome maturation, and receptor ligand sorting can then take place. I've provided a couple of links to animations showing the process of clathrin-mediated endocytosis. One shows a general overview, and the other provides more detailed information about the other protein players involved, so be sure to check those out. It is a very cool process. This video has provided an overview of cell signaling, an introduction to the specific receptors and signal transduction messengers that are commonly used, as well as important mechanisms for signal termination. Next, we'll dig deeper into the details of signal transduction by delving into some specific and biologically relevant examples of cell signaling.